in meiosis. What is meiosis? Meiosis is a type of cell division in sexually reproducing organisms that reproduces or reduces the number of chromosomes in gametes, the sex cells or the egg cells, and the sperm cells. So, meiosis, it is a type of cell division that happens in sexually reproducing organisms. Sexually. So, gametes. These are the what? The sex cells, sperm cell, and the egg cell. So this is what happens during the reproduction of sperm cell and egg cell. So it results in the formation of the gametes, which have half the number of chromosomes as the original cell. Meiosis takes a cell with a full set of chromosomes, the two sets, one from each parent, one from the mother and one from the father, and it divides into 4, not 2 but 4 cells, each with only set of chromosomes. This halving of chromosome cell number is very important for sexual reproduction because when these two gametes or the sperm cell and the egg cell combine just during the fertilization, they create offspring with the correct number of chromosome. So, one set from each parent. Okay? So, meiosis results in the formation of 4 daughter cells at the end of the process. So, these 4 daughter cells are unique because they are not genetically identical to each other. They have different combinations of genetic information due to the shuffling of genes and chromosomes crossover events during meiosis. The key feature of daughter cells produced by meiosis is that they are haploid. What is the meaning of a haploid? Haploid, meaning they have only one complete set of chromosomes. This is essential for sexual reproduction because when the two haploid gametes, the sperm and the egg cell, fuse during fertilization, they create a diploid zygote with the correct number of chromosomes. So, haploid refers to conditions in which a cell or organism has only one complete set of chromosomes. So, each of the 4 daughter cells are haploid. In humans and many animals, this means having half the typical number of chromosomes found in the diploid cell. So, in diploid cell, there are 2 sets of chromosomes, diba? One from each parent. The most common example of haploid cells are in the humans. The sperm cell and the egg cells are haploid. The sperm cell are produced in the testes, and the eggs are produced in the ovaries. So each of these reproductive cells is haploid because they contain only one set of the 23 chromosomes in humans. So 23. Each of the sperm cells and the egg cell, they only have 23 chromosomes. Why? Because when you combine sperm cell with 26 chromosomes and egg cell with 23 chromosomes in the fertilization, so 23 plus 23 equals 46. So 23 means haploid, 46 means diploid. So when they fertilize the egg and the sperm cell, the zygote is now diploid, having 46 chromosomes. Okay? So remember that. Haploid, only 23 chromosomes. Diploid, is complete with 46 chromosomes. The egg cells and the sperm cells only have 23 chromosomes, hence they are called haploid. Okay? So this one is the overview of the meiosis. So the meiosis involves two rounds of cell division, the meiosis 1 and the meiosis 2. So in meiosis 1, the homologous chromosomes, one from each parent, are separated into different cells, reducing the number of chromosomes into half. While in meiosis 2, it is similar to mitosis. The sister chromatids of each chromosomes are separated into separate cells. Okay? So, step 1, the chromosomes duplicate. Step 2, homologous chromosomes separate. And step 3, the sister chromatids separate. Okay? So, during the prophase 1, in the meiosis 1. So, meiosis 1 is the first division. It involves the separation of the homologous chromosomes. So, during the prophase 1, homologous chromosomes pair up and may exchange genetic materials in the process called crossing over. It is the introducing genetic diversity. In metaphase 1, these paired homologous chromosomes line up along the cell's equator. During anaphase 1, the homologous chromosomes are pulled apart and moved to opposite ends of the cell. In telophase 1, two haploid daughter cells are formed, each containing one set of chromosomes. So these chromosomes are still in duplicated form, each with two sister chromatids. The next phase is the meiosis 2. What happens in this one? It involves the separation of sister chromatids. So in prophase 2, it is a brief preparation phase. The nuclear envelope may reform around the chromosomes. Metaphase 2 aligns the duplicated chromosomes along the equator of the two haploid daughter cells. Anaphase 2, the sister chromatids are finally pulled up and moved to opposite ends of the cell, while in telophase 2 completes the meiosis 2, resulting in a total of 4. So you can see here, there are already four haploid cells, each with a unique combination of genetic material. This process is vital for sexual reproduction and genetic diversity. So you can see that each of these 4 haploid cells formed, they have different or unique combination of genetic material. Okay, so let us start with the meiosis 1. It is called the reduction division because it reduces the chromosome number into half. Okay, so let us consider in humans where the diploid number of chromosomes is 46 or 2n. 2n. N is 23. So 2 times 23 is 46. So in meiosis 1, a cell with 46 chromosomes or 2n undergoes the reduction division. During the process, the homologous pairs of chromosomes, one set from the mother and one from the father, are separated. As a result, what happened? Two daughter cells are formed, each containing 23 chromosomes or N, which is half of the parent cell's chromosome number. These two daughter cells are now haploid and ready to go through meiosis 2, which further separates the sister chromatids to produce a total number or a total of 4 haploid gametes. So in meiosis 1, it is the reduction in chromosome number, leading to haploid daughter cells, and this is essential for, as I repeat, sexual reproduction and genetic diversity. So these are the 4 stages of meiosis 1. So as you can see, we have prophase 1, metaphase 1, anaphase 1, telophase 1, and the cytokinesis. So, what happens during the prophase 1? So, at the beginning, pairs of similar chromosomes come together. Each chromosome looks like a long string made of two parts. When they are paired up, they create a structure called a tetrad. So when they paired, it becomes tetrad. Okay? Which is like four strings slide up side by side, a kind of four friends holding hands. So that is called tetrad. So during this time, the two strings in each pair can swap pieces of themselves. This swapping is called the crossing over. And it mixes up the genetic information. It's like if you and your friends traded some of your toys or makeup or snacks to share and change things up. So as prophase 1 goes on, the chromosomes get all coiled up tightly, and the special structure called the spindle are forming. So this covering around spindle fibers, the covering around the center of the cell breaks apart, and the tetrads held by the spindle strings move towards the middle of the cell. Okay? Next, the prophase 1 is divided into 5 stages. So we have leptonema, zygonema, pachinema, diplonema, and the diakinesis. Okay? So, what happened during these stages of prophase 1? So, in zygonema, homologous chromosomes start coming together as pairs. These pairs, one from each parent, are called homologous pairs. Each pair line up with its partner aligning gene by gene. This alignment is called synapsis. So when they align, the alignment is called synapsis. Next one is the pachinema. First is the zygonema. The second, or zygote. Zygote and zygonema are the same. Pachinema or pachytene, what happens in this pachinema or pachytene? Within the paired homologous chromosomes, a process called the crossing over occurs. So, in pachinema, the crossing over happens. It involves the exchange of genetic material. So, when we say crossing over, it exchanges the genetic material, the segments of DNA between two non-sister chromatids of the homologous chromosomes. Crossing over results in the genetic recombination, creating new combinations of genetic information. Next is the diplonema or diplotene. After crossing over, the homologous chromosomes begin to separate slightly, but they remain connected at points called the chiasma. So the chiasma, where the crossing over occurs. This allows for flexibility in the pairing. And the last one is the diakinesis, the final substage of prophase 1, where the chromosomes continue to condense, becoming more tightly coiled. The nuclear envelope begins to break down and spindle fiber starts to form around the chromosomes. Next one, we have the metaphase 1. So in metaphase 1, the paired chromosomes groups or the tetrads are lined up in the center of the cell. And sila sa center. Okay? The tiny rope structures or the spindle microtubules are attached in the middle of each chromosome pair. It's like these tetrads having ropes tied to them at their center. So in each tetrad, the two similar chromosomes, one from the mother and one from the father, are connected at places where they swapped information. So this swapping of information is called the crossing over. It is like they are holding their hands. Okay? So the centromeres line up at the metaphase plate. The homologous pair can be seen at the center, facing both sides of the poles. Next one is the anaphase 1. So in anaphase 1, the chromosome starts moving towards the two opposite ends of the cell. Okay? So, what is the difference of this anaphase from mitosis? Unlike the regular cell division or the mitosis, the twin-like parts of each chromosome or the sister chromatids, they stuck together. They don't separate. Look at the homologous chromosomes. So, they are tetrad, diba? Apat. So, look at the combinations or the genetic material. Okay? So, they don't separate. It's like a tug of war set up where the chromosome pairs are ready to be pulled towards the opposite ends of the cell. So, in anaphase 1, the chromosomes is moving to the opposite ends of the cell. So these sister chromatids, they don't separate. They stuck together. Only the groups of paired chromosomes or the tetrads, which are like sets of friends, split up. So in the picture, you can see that the three pairs of doubled chromosomes are now on the move, heading to each end of the cell. So it's like these pairs of chromosomes are going to the different sides of the cell. Okay, and the last one for meiosis 1 is the telophase 1 and cytokinesis. Okay? So in telophase 1, the chromosomes are what? Reaching at the ends of the cell. At the end of the cell, there's now a group of chromosomes, but they are still in pairs, like having twins in each group. So, look at the genetic diversity. Lahi-lahi siya, oh. Color. Okay? Different combinations. They are not the same. At the same time as telophase 1, the cell starts to split up into smaller cells, and this process is called cytokinesis. The division creates two new cells, and each of them has half the number of chromosomes as the original cells. 23, 23. However, these centrosomes or these chromosomes are still in pairs with two parts connected. These new cells are called haploid cells because they only have one set of chromosome. So after telophase 1, in some creatures, there's a pause called the interphase before they move to the meiosis 2. In others, they go straight to telophase 1 to meiosis from telophase 1 to meiosis 2. In both cases, the chromosomes stay the same and they were after telophase 1 and don't duplicate. This helps create cells with different genetic combinations in which it is important to produce diverse offspring. During metaphase 2, the sister chromatids within the two daughter cells separate, forming 4 new haploid gametes. It's directly undergo cell division. We can see here an illustration about meiosis 2. It involves prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So this meiosis 2 is like mitosis. Okay? So in prophase 2, a structure called the spindle forms inside of the cell. The spindle is like a set of strings that will help move the chromosomes. The chromosomes, which are still in pairs from the previous division, starts moving towards the middle of the cell, getting ready for the next round of cell division. So they are lining. Spindle fibers are formed and the chromosomes are lining. Okay? So what is the focus of prophase 2? It is for preparing the chromosomes for the next cell division. And that is the metaphase 2. The chromosomes lined up neatly in the middle of the cell. So look at the cells lining up the equator. Just like they do in the mitosis. Okay? Next is the anaphase 2. The same. But the same. The division. With the same with mitosis. In anaphase 2, the copies of DNA or the chromatids that were stuck together like twins in the cell split apart, and each copy goes to a different end of the cell. So they move away from each other towards opposite ends of the cell. While in telophase 2, little nuclei form at the end of the cell. The cell divides into 4 smaller cells. These new cells each have half the usual number of chromosomes, which means they are now single chromosomes, not pairs. So you end up with 4 daughter cells. 4 haploid daughter cells. Okay? So this is the entire process of meiosis. 2 is lot like mitosis, but it happens in cells with only one set of duplicated chromosomes. Okay? So as a summary, we can say that in prophase 2, spindle fibers form and push the chromosomes to the middle of the cell. While in metaphase 2, the chromosomes lined up in the middle, just like in mitosis, with their kinetochores or handles pointing in opposite direction. While in anaphase 2, the handles or the centromere splits, separating the chromosomes, which were copies of each other, into individual chromosomes. These individual chromosomes move to opposite ends of the cell. And in telophase 2, nuclei form at both ends of the cell. At the same time, the cell divides into 4 smaller cells. So these 4 new cells have only one set of chromosomes, making them haploid, and they're no longer paired like in the beginning. So you end up with the 4 daughter cells, each with single chromosomes. So here, we can see a Venn diagram that compares the mitosis and meiosis. Okay, meiosis and meiosis. Difference between them. Okay? So what is now the difference between mitosis and meiosis? Aside from the fact that mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells. Let us discuss the purpose of mitosis. So, mitosis occurs in somatic body cells, and it is responsible for growth, tissue repair, and replacement of old damaged cells. It results into two identical daughter cells, each with the same number of chromosomes as the parent cells. While in meiosis, it is a specialized form of division that occurs only in germ cells or the sperm cells and egg cells and is responsible for sexual reproduction. It reduces the chromosome number by half, resulting in haploid daughter cells. Okay? How about genetic variation? So in mitosis, it does not create genetic diversity as the daughter cells are genetically identical to the parent cell. While in meiosis, it creates genetic diversity through processes like crossing over or the exchange of genetic material between the homologous chromosomes and a random assortment of chromosomes during meiosis 1, which results to a genetically unique offspring. So, as an example for that are the identical twins. Diba? Identical twins are just like you're having your normal brother and sister because you have different set of genes. You form from a single ova, but there are two sperms fertilize the ova, so it divides into two. So you have these different sets of chromosomes. Lahi jud ang itsura. Okay? Ninyo. So this one is the illustration of the differences between mitosis and meiosis. So you just read the difference. Okay? Let us view a video.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has two sets of chromosomes. In humans, that's including one set of 23 chromosomes from mom and one set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away. The chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make two new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form two cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with two identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells, gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, that ends of the topic meiosis and their differences. So I stand corrected ha, kanina we are discussing the genetic diversity and the example is the twins. So we have two types of twins, the identical and the fraternal twins. So in identical, you have one sperm and one egg and it divides to form two organisms. So the organism have the same copy of the genetic material, thus they are physically the same. While the fraternal twins is just like having another siblings. So you have the same, you have one ova and you have two sperm cells that fertilize the ova. So each of the person is different from each other. Mostly, the fraternal twins, they can have male and female, while the identical twins must have both male or both female. Okay? So, the fraternal twins have different set of genes. It's like you have your sisters or your brothers, even though you are twins, but you are genetically different. Okay? So that ends about the meiosis, division, and the differences between the mitosis and the meiosis.